That's time. Good morning, good afternoon, good evening, everybody. Welcome at this webinar, Exploring the Microbiome, Part 2, following the first one, presented by our CSO, Françoise Levacon. Today, we will have a presentation from my colleagues, Diego, Sébastien, on Bioinformatics and Data Science from raw data. Conclusion. The agenda will be first a quick overview of Biofortis, who we are, what we do. Then three parts, bioinformatics tools, statistics and machine learning tools. And take-home message, this is a presentation of around 25 minutes with a session of questions. You can ask questions by the tool, the GoToWebinar tool in the questions part. So start. Biofortis, who we have, few minutes to remind you that we are very proud to be part of a family-owned group, Institut Mérieux, founded in 1897, following the footsteps of Louis Pasteur. Institut Mérieux, that's 18,000 people in the world, 3 billion euro of turnover, and three main corporations. Bioinformatics, leader in the world in the diagnosis, transgene dedicated to solutions for immunotherapy, vaccinal oncology, and Mérieux NutriScience. So Biofortis, here we are a part of this corporation. What's our job? Our job is to use human, real-life animal, but human trials data in real-life conditions to do data acquisition, bioanalysis, and to take in charge of data delivery, computing, valorizing this investment and return investments to improve health and well-being in the populations. Our job, that's to manage, to conduct clinical trials, to output data and analyze data by the way of central lab activities, and with a big focus on microbiome. We speak from human to data, from science to market. We are in the world with three main locations. In France, where we are here in Nantes, with two clinical investigation centers, private ones, and one central lab platform. And we have also a clinical investigation center and affiliates in Chicago, USA, working in several health fields like metabolism, weight management, oncology, oxidation. And since 2002, more than 500 projects managed by the team, and more than 250 clinical trials. French Biofortis Task Force, that's more than 80 collaborators this year, with a clinical business unit, with a lab and microbiome business unit, with a support function unit, and with an R&D business unit. 20 years of patient health and centricity for coordination, investigation, controlling, and regulatory aspects, for equipment, logistic, bioanalysis to help centers to include and enroll subjects and patients. Also, IT tools and quality management system, a strong one, applying the ICH GCPs and certified ISO norm. 35 people for clinical, 15 for lab and microbiome, 25 for support, and 5 for our own R&D business unit. I'm very pleased and proud to announce and introduce my two colleagues, Sébastien. Sébastien is the CTO, Chief Technological Officer of Biofortis, Head of Statistics and Bioinformatics, and involved in the lab part, in charge of the lab analysis, dedicated to research also and development projects. He initiated and applied within Biofortis, developed in Biofortis our dry and IT tools and helped to implement next generation sequencing in Biofortis laboratory, trained by science, bioinformatics, and have a degree in statistical methodology. Thanks a lot, Sébastien, to present us today this session. And Diego Tomassi recently joined Biofortis. Welcome, Diego. Diego is data scientist senior with a degree in bioengineering and a PhD in engineering. Was professor and past experience as a researcher in academia and including in National Council for Scientific and Technical Research of Argentina. That's your time, guys. Sébastien, next slide is your one. And please remind the first part of this webinar. See you later.
Thanks a lot, Stéphane, for the introduction. First of all, just to give you a quick reminder about the take-home messages of the first part of our webinar, exploring the microbiome, done by our colleague Françoise Levacon. So for clinical research, microbiome studies need an appropriate experimental, methodological, and statistical design. It is recommended to associate microbiome outcome and biological markers, and to conduct clinical trials investigating microbiome, it is necessary to get in touch with an expert partner. So we saw that exploring the microbiome can be done by monitoring different biological biomarkers using different approaches, including next generation sequencing. In the first part of our talk, we will focus mainly on the investigation of microbiome using NGS technologies, insisting on associated bioinformatics with an overview of approaches, challenges, and outputs. Decreasing costs associated with sequencing, quickly evolving technologies. Today, we are in front of more and more data generated by NGS platforms, which represents a huge challenge for their analysis. The title of our talk is Bioinformatics and Data Science from Raw Data to Conclusions. But the main message we would like to have today is to show how it is possible to accompany you to start from this complex initial data to highlight a simple scientific interpretation and message. Now, just to give you an overview of the complete workflow, once we collected the samples and run the sequencing, we have a picture of the biological world. But we still need complex analytics to extract the answers to our scientific questions from the raw data. The output from the sequencers is a huge volume of noisy data that it is not friendly to statistical analysis. The role of bioinformatic tools is to translate it into a format that can be understood with statistical tools without introducing technical bias. It then integrates the microbiome information with clinical data and other covariates in order to to get an answer to our question. So now, to lead microbiome investigation, 3 different components are essential when we are using NGS data. First of all, of course, lab capabilities and scientific expertise to generate data of quality and advice for design and biological or scientific interpretation of data. Secondly, a multidisciplinary data analytics team for the analysis of generated data, including IT for infrastructure administration, data management for the cleaning and the centralization of data, bioinformaticians to generate intelligible data for statisticians and data scientists. And finally, we also need an infrastructure able to store and back up the important volumetry of data. We also need computational resources to allow the intensive calculus in bioinformatic pipelines in a reasonable time, and also large pipes between this infrastructure. When we are investigating microbiome using NGS, 2 different approaches can be considered starting from an ecosystem. The first one is metabarcoding, also known as Amplicon sequencing, which targets specific regions of DNA, for instance, a region of the 16S rRNA gene, which is found in all bacteria, or the ITS sequencing for fungi. Bioinformatic pipeline generate taxonomic profiles, answering the question, who is there? Several thousands of reads are generated with this approach. The second approach, called shotgun metagenomics, whose aim is to break DNA in small pieces and sequence them. The sequencing depth is here much more important, up to several tens of millions of reads, depending on the biological matrices and associated host contamination and scientific questions. Bioinformatic analysis provide taxonomic and also functional profiles, adding information about the present genes. 2 biological questions can be answered here. Who is there and what can they do? In metabarcoding, we are interested in the taxonomic composition of the sample communities. The output from the bioinformatic processing is a matrix of counts that represents abundances. It is the most economic approach, but it cannot reveal the full ecosystem as it is a targeted method. There exist several pipelines proposed to do this, such as Mother or Shime, including different steps, quality control, cleaning, merging of amplicons, or clustering or denoising. However, every step requires setting suitable algorithm and parameters, and the choice made can affect an inferred ecosystem in terms of sensitivity and specificity. 2 main bioinformatic approaches can be considered in metabarcoding analysis for representative sequence selection. We are speaking about clustering to OTU or denoising to ASV. In the approach based on operational taxonomic units, OTU, similar sequences within a predefined threshold are clustered into the same group and then mapped to a reference database. Since each cluster includes some variation, taxa assignment is less sensitive to technical variation, but can also miss some variation that it is biologically meaningful. On the other end, we have Amplicon sequence variance, ASV, which ideally searches for an exact match between sequences. This provides improved sensitivity and specificity and helps to merge data across studies. Nevertheless, this approach needs to protect further against sequencing errors, sometimes resulting in an increased loss of read. While metabarcoding is useful to characterize specific type of microorganisms in a given experiment, like for instance bacterial genus, shotgun metagenomics provide a global overview of the microbiome composition and its functional properties. We can have access to gene annotations such as gene ontologies, metabolic pathways. The increased resolution up to species or strain level comes at the price of a much bigger volume of data needed to get enough usable reads. Beyond quality control and cleaning of reads, the first step of the bioinformatics pipeline is typically to remove genomic material from the host in order to obtain acceptable signal levels. We can have, according to the biological matrix of interest, more than 80% of host DNA, and as a consequence, only a small fraction of reads possible for microbiome investigation. So shortly, metagenomic sequencing data are typically analyzed either by de novo assembly or by comparing reads individually to reference databases. The mapping-based approach is faster and more economical, but with de novo assembly, we can identify previously unknown members of microbiota. However, assembly requires substantial sequencing depth, then assembly-based methods are typically restricted to the genomic reconstruction of highly abundant microbiome members. So, as you can see, this table extracted from an article from Clisson in 2017, plethora of tools and algorithms are available in the literature, and even for each step of a bioinformatics pipeline. Up to now, there is no gold standards. Tools and algorithms evolve quickly, and we can find a lot of benchmarks in the literature. Choice of bioinformatics pipeline also depend on the initial scientific question. Key points to consider to keep the same tools, parameters, and databases through your projects. So now, starting from these first results provided by bioinformatics pipelines, that is to say taxonomic or functional profiles, how I can go deeper in the interpretation of my results. The first step is to simplify the messages contained in this high-dimensional data by using visualization in order to make them more intelligible and comprehensible. The idea is to create graphical representations that give you biological insights without to be too complex. In addition, when we are interested in the variation of microorganisms in a single sample, alpha diversity indices are calculated and can be defined and measured through 3 different dimensions. The first one is called richness and refers to a number of species. The second one is evenness, also known as equitability, and refers to the proportion between species. And the last one is disparity, which refers to the phylogeny, to the genetic distance between samples. One other interest to assess the is to assess the variation of microorganism communities between samples, which is called beta diversity. We are in front of different measures of beta diversity that can be used, such as, for instance, Jacquard, which is based on the presence or absence of taxa, Bray-Curtis, based on abundance of taxa, and Unifrac and its 2 variants, which is based on phylogeny and second distances. Finally, beyond alpha diversity or beta diversity, several means of increasing the power of studies can be to stratify the population according, for instance, to enterotype, low gene count or high gene count, or to focus the analysis on certain bioindicator species such as Akkermansia municiphala or Faecalibacterium prosnitzi, which are currently considered 2 of the main beneficial bacteria in the gut. To facilitate the visualization of this high-dimensional data, we have developed internally an interactive web platform from our customers, which is called EasyBiom. This platform is hosted on a server approved for data hosting in order to guarantee the security, the integrity, and the confidentiality. Descriptive statistics and graphical representations are proposed in the EasyBiom application. And in addition, some classification methods are also implemented, such as clustering, principal coordinates analysis, etc. Developments are currently underway to provide new tools for our customers. So now I can let the end to my colleague Diego to give you an overview of statistics and machine learning tools.
So thanks a lot, Sébastien. So the output from the bioinformatics pipelines help us to understand who is there in the microbial sample and what they can potentially do. But in clinical trials, we want to go further and we want to study the interplay between the microbiome composition and some outcomes and covariates. So for instance, is there a change in the microbiome profile due to a treatment? And if so, who are the species that show that differential effect? And for these questions, we need to rely on statistics in order to have guarantees in our findings. So we need tools first for visualization and exploratory analysis, like detecting outliers or clusters. We also need tools to test for overall association between the microbiome composition and the outcome of interest. And in doing so, we typically need some other covariate to adjust, and we can deal with repeated measurements across time. And if we find some significant effect, we want to study in detail what are the taxa responsible for the effect. And to do so, we need to test a large number of hypotheses at the same time. And so we really want to have maximal power for detecting the effect, but at the same time, we want to control the rate of false discoveries. So all of these are rather typical problems in statistics, but microbiome data has some very typical features that make these tasks still very challenging. So first, microbiome data is high dimensional and it's count data with lots of zeros. And it means that we really cannot apply the most standard statistical tools to handle this data. So microbiome data is also hierarchical if we focus on taxonomic data, and this requires some structural coherence when we analyze this data. So phylogenetic relationship can also be important, but most important perhaps is the fact that we always work with relative abundance, and this means that microbiome data is indeed compositional, and this implies that we need to take the full vector of measurements into account at the same time. So we have multivariate data with some specific constraints due to compositionality, and this is very challenging. And on top of this, we can have some extra challenges due to the design of the experiment. Just to show a simple snapshot of this complexity, and to keep in mind that we do not have access actually to the absolute abundances, we need to keep in mind that we work always with relative abundance. So if in the true ecosystem there is only one taxa that is changing its abundance due to some specific treatment, this change will translate into the change of relative abundance for all the rest of the taxa in the observation. So it means that it's actually very tricky to pick which is the single change that is really meaningful. And in addition, because of the hierarchy in taxonomic data, we can perform the statistical analysis at different taxonomic ranks. But the conclusions we obtain from the analysis should be coherent across the taxonomic ranks. And this is something that we cannot always guarantee with the standard statistical tools. So which are the tools we can use currently to deal with the analysis of microbiome data? So, well, in the visualization part, the most common tools are principal coordinate analysis and some non-metric variants of multidimensional scaling, coupled with some specific notion of distance like Bragg-Curtis or unifrac distances. And when using these tools, we need to keep in mind that the representation we get can be sensitive to the choice of distance we made, but maybe most important, these representations are indeed insensitive to the outcome of interest. So if in our study we are interested in 2 or more endpoints, with these tools we only have one representation that may not reveal a close relationship with the outcome. So to test for overall association, still a common tool is Permanova, which is a permutation procedure that tries to mimic the standard analysis of variance. And one main limitation of Permanova is that it really cannot handle covariates and repeated measurements. So we have more flexible tools for that, like kernel association tests with regression models. An example of that is Meerkat. And with these tools, we can accommodate a different notion of distance and also handle repeated measurements. But what we cannot do with these tools is to test for subsets of taxa being differentially expressed. So for doing this, we need to rely on some specific tools for differential abundance analysis. And we have some univariate tools that comes from standard statistics or also from RNA-seq analysis. And they are nice in the sense that we can fit complex design with them, but at the same time, they can be quite sensitive to the normalization of the data we use. And they can also be prone to false discoveries because of the univariate approach. So most recent approaches try to leverage the full compositional nature of the data so that we can reveal truthfully the real full ecosystem. But these new tools are still less developed, and sometimes we can have problems to fit a complex design. So do we have more advanced tools to deal with microbiome data analysis? Well, in general words, I think we can see an evolution of the tools we have, from standard statistics to some tools adapted from RNA-seq analysis to more modern tools trying to deal better with compositional nature, with phylogenetic relationship, which are intrinsic to metagenomic data, and also to include some ideas from machine learning. So for instance, for visualization, we can try to use more advanced embedding algorithms that give us a compressed representation that can pack more of the available information in just the first directions. And we can try to embed some additional information in the same plot, or we can use some sufficient reductions or canonical correlation, especially adapted to compositional data, in order to obtain some visualizations that have some information about the specific outcome of interest. Regarding association tests, we can try to improve on the power of kernel association tests by including some prior knowledge or some context-specific information in the regression model. And for differential abundant analysis, we can use more general models that even can account for different sampling fractions when collecting the sample, and a more general relationship between the taxa. So let me show you some examples of how these new tools can look like. So for instance, we can use new generalized matrix decomposition algorithms on graphs in order to obtain a plot that is able to combine the advantages of a standard principal coordinate analysis in clustering the data and about using some ecological meaningful distance with some features of standard PCA, like we find in a standard B-plot, that are nice in the sense that they give us some information about the relevance of each variable, which are taxa in this case. So we can combine both approaches with these new tools, and indeed we can exploit this type of tools to encode for and to take into account some other interesting priors. And regarding outcome-specific representations, we can actually exploit some theoretical link between a wide family of models and some efficient representation. So for instance, we can pick the models inside the Lefse analysis, and beyond running the differential analysis test, we can use the same information to do some visualization plots on the data. We can do the same with more general models, like coming from RNA-seq models, and we can do the same even with much more general models, which are functional extensions of exponential families that are used in machine learning. And all of them will give us a representation encoded by the parameters that is able to reveal some output-specific information. And this is nice because it allows us to have a connection between the exploratory analysis and the inferential analysis, and we can do this even for continuous variables and for multivariate outcomes too. So regarding testing for overall association, I think we should take into consideration that we typically use these tools with default settings for a standard with the standard kernels derived from unifrag and break-artist distances. But the power of these tests actually depends strongly on the kernel we choose. So we can try to learn adaptively a kernel according to our specific experiment. So for instance, we can try to put there some prior information, some results from network analysis for subcommunity detection, and a lot of tools that we can borrow from machine learning, and to include the results in this statistical test that is already available. Regarding differential abundant analysis, as I said before, there is a strong trend in using some approaches for compositional data analysis, like compositional balances and differential ranks. But which is the best method? I think it's still not easy to, it's a question still not easy to answer, because it can depend on the design and also on the sample size. And even after having a set of selected taxa, we can be interested in ranking them according to the relevance to describe the outcome. And we can do that, for instance, using random forest. And as I said before, we can also use some ideas to do some adaptive control of fault discovery rates, taking advantage of the hierarchical nature of the data, so that actually we reduce the number of multiple comparisons, and we can preserve power better in doing that. So in summary, we have a lot of tools available. I think there is no gold standard for every scenario. So the best choice is always problem dependent, and actually there is no recommendation guidelines that covers inferential statistics during the analysis of microbiome data. So we really require expertise and a full comprehension of the full experiment design in order to pick the best method for analysis too. And to finish, just let me show you in what other ways we can use machine learning ideas to add value to clinical trials. And these opportunities come mostly from the increased flexibility we have with machine learning tools for do some operations like data integration and automatic extraction of context information, even beyond the known capability of machine learning in prediction. So we can take advantage of this increased flexibility in the full step of the design, just from the design of the experiment to the recruitment, to the statistical analysis, and even in some deeper post-hoc exploration of our results. So, for instance, whenever we want to combine some metagenomic data with some other metaomic data in order to try to understand better the interplay between the microbiome and the host environment, we can use machine learning tools in order to integrate the data. We can also use data from several studies to learn some complex models, which are autoencoded from deep learning, that are able to extract the main features and main relationships in the data, so that we have a compressed representation with the basic features. And then with new data, we can use this trained model to obtain an efficient representation, which will help us to do prediction or biomarker discovery. And machine learning can also help us to optimize clinical trials. For instance, we can build a recommendation system to improve the matching between volunteers and clinical study, or we can use some tools from machine learning to do some text mining in order to find automatically some confounders, some prior information that was not known in advance. We can even pack all of this information with realistic simulators of data in order to have a system that allows us to make decisions, but data-driven decisions during the design of the experiment. So we have really a large number of opportunities coming from these new ideas.
So thanks a lot, Diego, for your talk. So just to give you some last word concerning this presentation. So as take-home messages, we have focused today on a brief overview of the workflows, the challenges, the outputs to analyze, and also on the added value to the results you can obtain when you are interested, you are investigating microbiome, mainly in the framework of clinical research. As we try to give you this message also, no standard way exists to pick up your objectives, and it seems that we can be a real partner gathering all different expertises to adapt the methodology to your scientific hypothesis. And our company can today offer you a fully integrated or a modular solution from the design of the study to the lab operation and the valuation of your data. So thanks a lot for your attention, and we are available with Diego if you have any questions. So sorry, I think we have spent more time than initially scheduled.
Thanks, many thanks, Sébastien and Diego, for this not shallow but deep clarification and explanation. We have indeed several questions. We will take in charge, but not all this one now, but after by written, please. I will choose 4 questions, for instance. The first one, which is, what do you think about bioinformatic tools aiming at obtaining functional profiles from metabarcoding results? For example, PCRUST. Can they replace the shotgun metagenomic approach? Sébastien, Diego?
Yes, so this kind of tools, I think we can use these tools as to have a first ID, a first picture of genes or functionality which are only predicted or inferred. But I think it can be difficult in first instance to infer functionality of a metagenome only starting from a small portion of their genome, or for instance, these tools also map if we are interested in bacteria, they map the stickiness RNA genes to homologous taxa for which we have completed the sequences genomes. In the literature, there are several benchmarks about these tools. You cited PyCrust, but I have in mind also Pifilin or also Tax4Fun. And the idea of this benchmark is also to evaluate the performance of this method versus real data generated from shotgun metagenomics. And what we can see in this benchmark is globally we have a better performance using these tools on genes related to housekeeping functions. That is to say, it is more difficult, for instance, to have access to other genes, maybe which are highly phylogenetic variability, etc. So, in summary, yes, it could be a good idea to have just a first screenshot of the functionality, but in any case, it cannot substitute at all the shotgun metagenomic approach to have a clear and more detailed and resolutive access to the functionality of the metagenomes.
Thanks for this complete answer, Sébastien. Just two other questions. One technical for statisticians. Why do you present PCA graph and not PCA, and as in other scientific paper in that field? Diego or Sébastien?
Okay, I think I can answer this. I think in applied science, when we use standard PCA, often we want to show at the same time some information about the variables, for instance, using a B-plot. But with the standard PCA, we cannot use a more meaningful notion of distance. So here we have ecological data, so the standard Euclidean distance is not really meaningful. So we really want some more meaningful notion of similarity between observations. So to encode that, we need to rely on some variants of multidimensional scaling, and that's why we use principal coordinate analysis, basically.
Thank you, Diego. Last question, Géraldine, Sylvie, we will send you our answers after. The first one was Manfred. Hi, Manfred. I hope you are well. Thanks for your question. That's the question. Functional metagenomics. Are current tools sufficient to determine the still unknown functions of 40% of bacterial proteins? I can repeat that if you want.
Yes, if you can repeat.
So, the question, are the current tools sufficient to determine the still unknown functions of 40% of bacterial proteins? I don't know if there are 40% of unknown functions of bacterial proteins. Perhaps we do not have the answer today.
It is difficult for me to give you to quantify this, but of course, one of the main challenges when we are conducting shotgun metagenomics is what we call the bacterial dark DNA. That is to say, the DNA for which we have not yet any annotations in the public databases. So I cannot quantify this, but as far as I saw in the literature, there are some new tools which use machine learning just to try to infer also functions for this dark DNA. But of course, we are speaking about in the presentation that we lost a lot of reads according to the host decontamination. But of course, at the end, after bioinformatic analysis, we can have also a room of improvement when we are in front of this dark material or dark DNA.
Thank you, Sébastien. Just a last quick question from the attendee, from Sylvie. What is the pipeline used in shotgun Biofortis analysis? You choose Kraken, Keiju, others?
Yes, for the moment, we are currently, so we have our current pipeline use centrifuge and human tool for taxonomic classification and functional analysis. But we are currently working on possibly alternatives for two main reasons. Is that for the moment we are using more mapped approaches, what I call mapped-based approaches. That is to say, we mapped our reads against databases, and we would like first to increase our resolution, so up to the strain level analysis. So we are currently benchmarking so some other tools. And the second one is that with this pipeline, we would like also to speed up our time of analysis. So even if we have the possibility to use very huge computational resources, we have to speed up this. So this is why currently we have these two main tools, because the pipeline is an aggregation of different tools, but the two main are these two, centrifuge and human tool. But we are working on maybe on using others.
Thank you, thank you. So that's time now. Just the last slide to present you our last microbiota booklet on metabolism and diabetes. Please download it on our website or ask and request us. You can see on the picture on the right that all the team, thanks colleagues of Biofortis, are mobilized and online and on the floor in clinical centers and in our laboratory. We are available for any request. Have a nice Christmas break. Please share warm time with your family. Enjoy your gifts from Santa Paul, and take care. Bye-bye. Have a nice evening.
Bye-bye.